Okay, I think we'll start. Okay, good afternoon, everyone. It's my great honor to introduce our today's IAS distinguished speaker, Professor LIU Xiaogang from National University of Singapore. Professor Liu received his PhD in inorganic chemistry from Northwestern University in 2004. I think working with Professor Chad Merkin, right? And following that, he spent 2 years as a postdoc researcher in the Department of Material Science and Engineering at MIT before he joined in the Department of Chemistry at NUS in 2006. And he was then promoted to full professor in 2017. Well, now he's distinguished professor of Department of Chemistry at NUS. And he also holds joint appointment with Department of Material Science and Engineering and the Department of Surgery at NUS School of Medicine. And he has received many awards. Just name the most recent one. He received the RSC Centenary Prize in Chemistry and Communication last year. And his research interests are mostly the study of energy transfer in lens-like doped nanomaterials, and that's what we're going to hear most of it today. And he's very well known for the application of optical nanomaterials for biological applications as well as various optoelectronic devices. So without further ado, I think I'll pass the stage to Professor Liu. Professor Liu, the stage is yours.
Thank you. Yeah, first of all, I want to thank Haipeng for the very kind invitation. I recall my first time at the Hong Kong UST was almost 20 years ago. Yeah, when I first joined the AUS, and I visited here very briefly. So far, I really like what I see here. So I interact with some of the colleagues here and the students. Very exciting research going on here. So what I thought I would do today is perhaps introduce some of the recent work in lens-doped nanocrystals. I will perhaps highlight some of the applications. I think hopefully this research can resonate with some of your research. So first of all, I think that at the foremost, I want to, of course, thank the students and the postdocs behind all this. Otherwise, I wouldn't be here. So those are students. I think it's tremendous, smart students, hardworking students. Also, I want to acknowledge many of my collaborators. Some actually already moved to Hong Kong, and you see the Professor Xin Gang. And also, I see somewhere here. So Xin Gang moved to PolyU. I want to thank the collaborators. It's also, they're very important. Really push us to go after the bigger problems. They are also tremendous scientists themselves. Of course, the funding support from these funding agencies from Singapore. So my research focus on luminescence, labeling, imaging. So the most challenging questions in imaging, of course, is imaging resolution. How you can enhance the signal of biological species and then suppress the background noise. So that's I think that's the holy grail of the imaging community. So if you want to look at the cells, understand the cell, the mechanisms, you want to label this. Of course, two types of imaging approaches. One is label-free, using convention, for example, stimulated Raman. Or of course, the other one, most of researchers interested is actually using the luminescent probe. So those luminescent probes can really target the biological species you are interested in, so enhance the signal strength. Another challenge, I think, is for imaging is if you want to have this broad spectral images, so we call it multispectral or hyperspectral imaging, multi-wavelength imaging, is can you develop this kind of, we call this, portable, on chip. So the ultimate challenge is, can you develop miniaturized spectrometer that can couple to, for example, a smartphone, and then perform the imaging over a very broad spectrum range. So what you're seeing here is, so for example, if you want to perform space imaging, understand the spectral information that covers from radio wave, microwave, infrared, UV, X-ray, gamma rays. So that actually becomes the ultimate challenge because you think about the current strategies, it's actually using different detectors for a particular wavelength. If you want to detect X-ray, you have to use X-ray scope. UV, you have to use a UV telescope. So that's actually turns out it becomes very challenging if you go to the optical lab. Obviously, the complexity you need to set up. I just visit the optical lab. So it's kind of you look at the complexity. You need optical lenses, micro lenses. You need optical choppers. You need to also do the laser alignment. So those are quite a challenging issue here. Can we simplify this optical setup or reduce the number of components used for optical imaging? So one solution, perhaps we can go to these luminescent materials. We also call the frequency conversion materials. Those materials is already out there. We didn't create this. So it's already there long time ago. So this Nobel Prize already recognized this small organic dye GAP proteins. And then recently, of course, the quantum dots. So the capability of these fluorescent materials, you can see that, so they can precisely convert a particular excitation into specific wavelengths. So that's actually quite interesting because you really don't need to use a micro lens optic chopper to get this particular wavelengths. You can actually using the materials convert near-infrared light or X-rays into visible. You can get a very narrow emission output. So that's a benefit of using this lens, using these luminescent nanomaterials. For example, even small organic molecules, clusters, quantum dots, laser doped particles, and also some of these meta-organic frameworks or covalent organic frameworks, these are single crystals. Those are ideal materials, scintillators for X-ray imaging. So you have all these materials. Of course, the challenge remains is how you, so compared to conventional, this optical setup, these materials also face a challenge. The challenge is conversion efficiency. You talk to optical expert, they always ask you, the materials are quite interesting, but what about conversion efficiency? Because you're using a laser, optical setup give a pretty high conversion efficiency. Some of the materials, especially I focus on these laser doped particles, the photon up conversion, at the moment, the efficiency is still quite low. So, yeah, 2 years ago, so I'm very happy for the three researchers for their research in quantum dots winning the Nobel Prize. I think it's highlighting the nanomaterials has a potential for various applications, not just for labeling, but also I think the Nobel Prize Committee actually mentioned the LED applications of this beyond the fundamental research. So I think one interesting note is Louis Bruce, Alex Ekimov, they win the Nobel Prize because for the discovery of this quantum confinement effect. Monjiba Wendy, I believe, is for the improvement in chemical synthesis. So actually, that's very important because in the early days, I think most of the times, Nobel Prize always recognize something, the first discovery. But also, actually, Nobel Prize also recognize improvement in a certain research. So this is a highlight because his group actually discovered, how to make how to scale up the quantum dots in a biological friendly solvent. So that's actually lesson we learned. So I highlight this quantum dots, the laser doped particles. Also, so behaviors are quite similar in terms of the emission labeling potentials, but they are very quite a different class of materials in terms of the emission mechanism. Is very different from quantum dots that rely on this quantum confinement effect. So for laser doped particles, the emission comes from the emitters. Basically, it's actually the laser dopants within the host matrix. So it's because this laser dopants emission. So that also we actually can control the colors based on the controlling the energy transfer, the transition between energy, there's many energy levels. So for laser ions, the energy transitions of energy levels are enormous. For example, European has more than 3,000 energy levels. So actually for calculations, it actually become very challenging. But luckily, we have for many experimental results, we actually can now pretty confident to control the emission colors by adding different dopants or play with the dopant concentrations. Also, you actually can control the luminescence using hybrid materials. For example, by using small molecules coating on the particle surface, control the energy transfer dynamics, or using triplet states from the organic compounds to achieve luminescent tunings. Later, I will give some examples. So my research focused on this, the up conversion. So up conversion, I think for most of you are already familiar, anti-Stokes emission, Stokes emission. So this Stokes emission is excitation and emission, but they have some overlap. So actually, I call this a large overlaps. So this is obviously understandable. So you excite the molecules, some molecules absorb, and then some energy release, either dissipated as a heat, and then give the emission, or cause the emission wavelengths is actually shifted to the red. How to achieve the anti-Stokes emission? So early days, I think, for example, George Stokes, so he had some ideas. Perhaps, anti-Stokes emission is a thermal stimulated emissions. So you look at this simplified energy diagram here. So once you have the excited states, you need actual energy. Typically, the energies come from the heat, and then generate to the very higher excited states, then come down to ground states. So then the emission for the thermal energies typically is the KT is much less than 1 electron volts at room temperatures. So that means this type of anti-Stokes emission is very weak. It's actually invisible to naked eyes. So it's also sometimes it's actually very difficult to detect. So how to solve a problem if you really want to have a high efficiency anti-Stokes emission to solve the challenge? So these researchers discovered that using laser ions doped into crystalline structures, for example, this inorganic crystals, sodium yttrium fluoride, you actually can achieve pretty strong, how strong is actually visible to the naked eyes, up conversion emission. So the discovery of this photon up conversion was around 1966, 1967 by these three researchers. They actually met each other in St. Petersburg in International Chemical Conference, in the International, I think, Chemical Luminescence Conference in 1972. So they're really pioneer in the field. Why are we using this up conversion? So the primary reason is actually for biolabeling, as I mentioned, for image applications. So I just give you a few reasons why we're interested in this. Because for bioimaging, right, the first thing you think about is minimize the background noise. So if you use a near infrared light as an excitation, for example, 700 to 900. You can minimize the background absorption. So for biological environment, the first thing is water absorption, right? And then the tissue, skin, hemoglobin. So around these spectral excitations, you can really minimize the background absorption. So then enhance the signal. The other thing is actually if you're using near-infrared light as excitation, we talk about upconversion. Near-infrared light gives a much better penetration. So the penetration here, of course, here is showing the red excitation. So compared to the green excitation, okay, red has much better penetration than the green excitation. And also, these laser-doped particles, the photostability is very good because you can see here this is time-dependent photophysics studies showing that the particle luminescence is very, very consistent. Intensity almost remains essentially unaltered is because of the within these particles, you have many lens emitters. Unlike organic dyes of bleaching, the quantum dyes have the blinking problems. So laser-doped particles, very consistent emission over longer, very long time. So this actually gives the capability for in situ tracking. I will give you some examples using this technique. So this is the fundamental for upconversion, the principle of upconversion. I just want to quickly go through that. Materials, so typically we use sodium iodeum fluoride, doubled with YB as a sensitizer. TEM, ER is the activator. Sometimes we call it emitters. So all this energy transition correspond to these emission bands. You see the 4 emission peaks actually correspond to ER. So you have 4 different energy transitions. So you can match all these transition bands with the emission bands. The TM also, you have 3 emission peaks comes from these energy bands. So you can also tune the colors by varying the dopant concentration and also sometimes the concentration of the dopants. I want to highlight this. This is actually quite important work in the field. We published almost 15 years ago. We actually realized that, so this is quite interesting discovery. We realized using the lens at dopant, we actually can control the crystal phase. So this is a quite interesting study because you add some lens ions into the crystal structure. You can see we actually control the cubic phase to hexagonal phase by introducing the lens ions. So this is quite still, I think is a very interesting studies. Is because of this, we add lens ions, we introduce, we actually change the polarizability of the crystal field. Then we actually can achieve this the phase tuning here. So also I want to highlight this energy migration. So these laser-doped particles have many interesting optical properties. So energy migration is one very, very interesting properties. So energy migration means the energy absorbed by lens light sensitizer can pass to the emitters over a long distance. So this kind of energy, long-range energy transfer can only be done using lens lights, ions. So quantum dots cannot do that. Organic dye molecules cannot do that. So I was given a presentation, I think a professor from National Taiwan University. He actually showed very impressive results. Organic dyes perform this exciton migration. So they showed this 2 nanometer. So that's already almost a record for organic molecules. But actually for nanoparticles, I want to showcase here using this laser-doped particles, you can achieve energy migration over almost 20 nanometers. So the discovery, of course, from the bulk material to nanomaterial, I want to highlight the insight, the capability of using nanomaterials. So this paper is published, actually there's another paper much earlier than this paper, but it showcases that laser ions activated absorb the energy for example 80 nanometers, then transfer the sensitizer. You can see this is a random energy transfer process. So any migration can transfer to you, and to you, then you transfer, then go back and forth. So this is a competing process. But laser ions can do this. So by taking advantage of this phenomenon, we actually demonstrate using the energy migration, we can achieve photon up conversion for a broad range of laser ions. So this is a big deal because before this paper came out, before this work came out, for up conversion, we have a laser theories. Not all the lasers can achieve photon up conversion. Actually, only 3-4 laser ions can do that up conversion. But we actually extend the scope of photon up conversion to this, so the TBEU, DYSM. So we extend it now at least 7 laser ions can achieve the photon up conversion. I want to quickly highlight why this nanostructure is important. So because without this laser, these core shell structures, we cannot achieve this energy migration. So conventional discovery actually is actually using the bulk materials. But using this bulk materials without the core shell design, so the energy transfer sometimes is surprised by this solvent quenching. So we're actually taking advantage of the gadolinium ions. So that's the energy migrator. You can see the skin here. Takes the excitation energy absorbed by YB and then transfer TM. Then through the GD lattice. So the GD is within the core shell structure and then transfer to the emitter here. So why the GD can absorb the excitation energy and then pass to another GD through the sublattice is actually, if you look at the energy level here, so they don't have many intermediate states like TM and YB. So the energy gap here is about 4 electron volts. So 4 electron volts is very big. So it means even water, this vibration will not interfere with the energy transfer. So that's why the GD energy migration using the GD. GD is a magic. I think it's a magic element. So if you look at we try to identify the core shell structure, but this is a very good high resolution TEM image. We can even see the sodium ion. This little spot right here, but we cannot see the core shell interface because laser ions are very similar ionic radius. So you essentially don't see anything here. But using this elemental mapping, we actually can clearly see this is core shell structures. So also I want to highlight this very unique mentioned energy migration. So 3 most important unique properties. So I just mentioned energy migration using laser ion. That's the second. There's pulse duration sensitive color tuning, the second properties. Later I mentioned another one is photon avalanche. So for this work, what we have achieved is, so we want to tune the particle luminescence without changing the particle composition. So this is actually very challenging. So for quantum dots, if you want to tune the color, typically you have to change the particle size. But that means you go back to the lab, you synthesize the materials, and then you keep go back and forth. So the question is, can you make some materials just one with a fixed composition, but upon excitation, upon changing the external stimulation, for example, the pulse duration, pulse frequency, can you change the color? So that's the fundamental question we have. So it turns out that we can do that. So Landon, a very good student, so he came up with this multi-core shell structures. So it's a complicated design here. You can see originally the core particle is actually spherical shape, but upon this multiple synthesis, the particle is elongated. But it still is a core shell structures. I think some of the minor phase transitions, but it still is a core shell structure. Every layer has a certain combination of laser ions. So it turns out the design is very, very important. So how he come up with this design, I still, I often talk to him about this wonderful design here. Let's see the results you can see here. So the particles disperse in the solution. Then we use 3 different laser sources. So the top one is 980 nanometer long pulse. The bottom one is 980 nanometer short pulse. The middle one is AOA CW laser. So you can see laser source is different. The excitation condition is different. They give us complete different colors. So this is quite amazing. So at that time, so it turns out that we can actually using this core shell structure control the energy transfer rate between different laser ions. So we can even achieve multi-color tuning, even achieve the white colors using these designs. So that's the mechanism here. Looks very complicated, but let me summarize what we landed from this materials is. It turns out that different, for example, using 80 nanometers. So 808 nanometer excitation, the blue emission is actually come from the TM. Okay, so on the 980 nanometer excitation, the red emission is actually controlled by using the CE ions. So you can see the emissions come from different processes. One is 2 photon emission, the other is 3 photon emissions. So using the CE ion, we actually really can control the energy transfer pathways to achieve this color tunings. So what is useful? Once we have this material, so one demonstration is we demonstrate we can actually achieve this volumetric 3D display in full color. So it means we're using the laser scanning. We can achieve the build all this wonderful 3-color, multiple colors. So compared to this control experiment, if you just simply mix RGB color emitting nanocrystals, so what we have is we have the saturation colors. This is understandable because all the colors are saturated. We cannot control. If the particles are too close to each other, we actually get a white color. But using our materials, so we control the pulse duration, we can change the color. So this is one capabilities, actually using this pulse duration sensitive materials. So actually, after publication, I said the editor actually invited Lennon to roll this article, expected unexpected. So I want to share with some of the students here is the importance. As Lennon he had ideas, but we have to use a pulse laser. But that time, my group was very poor. We cannot afford a pulse laser. This pulse laser costs not that much, $10,000. So Lennon, so he made this. This cost $100. Okay, so you say basically this is homemade optic chopper. So because pulse laser, right? So this is very simple. So he tested his idea. So using the simple the designs worked. So then he came to my office. I said, well, we cannot publish a paper using this kind of design. So we bought the pulse laser later on. So I think this is very important, showing that the student, proof of concept experiment. It doesn't need a multi-million equipment. If you have a simple design, if you can prove your concept, then that's very good. It can save some, research grant. So I highlight some of these very fundamental properties, especially energy migration, this pulse duration, excited color tuning. So very unique capabilities from laser doped particles. So if I look back over the past 20 years since I joined NUS, I believe the field already evolved into many different fields. It's not really the chemistry or material science. It's a very broad range of, I would call this cross-disciplinary. Now, of course, including the AI nowadays. So this has become really the overlapping of different research field, biological field, photocatalysis, super resolution. So many capabilities you actually can use in these laser doped particles. So I want to highlight some of the interesting application using this laser doped particles. For fundamental study, if you're interested in this luminescent transfer FRAG, certainly you can consider using these particles because you can treat the particles as an energy donor. You can study energy transfer from the particles to this organic dye, very, very small right here. So energy transfer, how you transfer the energy, how you enhance the efficiency. So normally for FRAG, energy transfer is distance dependent. It's less than 10 nanometers. But if your particle have 20, 30 nanometers, how you transfer energy from this side of the particles to the organic dye is across 30 nanometers. Obviously, it's not enough. It's a distance is too long. So you can take advantage of laser ion, as I mentioned, energy migrations shown here. We show that using the energy hopping. So energy migration is more like an energy hopping through the GD lattice, okay? And then transfer emitters. So if I cannot, 10 nanometer transfer is 20 nanometers too far for FRET, but I can use lens ions as intermediate migrators. So we are showing that this is simulations. So without energy migration, the critical energy transfer distance for FRET is basically 4 or 5 nanometers. But using our designs, you can see we can extend it to almost 20 nanometers. We actually we validated this hypothesis using experimental data. So we actually using the core shell structure control the migration distance. Of course, another capability is you can using the particles study this triplet energy transfer process. You can study hybrid material systems. So, for example, a few years ago, I worked with Arkeshay group from University of Cambridge. We demonstrated that we actually can achieve the direct triplet generation using hybrid materials, lens doped particles with this very popular organic highly conjugate molecules. For example, the tetrasin rubrins. So with the lens nanoparticles, we actually can enhance the triplet absorptions based on the data shown here. It's quite interesting. We can also enable direct triplet generation. So I want to highlight this interesting studies. So it turns out this direct triplet generation is associated with unpaired electrons. Okay, it's not so conventional. There's also debate. It's a triplet generation. You can actually use in heavy atoms, or you change the spin orbital couplings, the energy mappings, the splittings. So from our experiment, we actually did a series studies across the lens series. It turns out that the number of unpaired electrons plays a significant role here. So if you look at the GD, LU obviously is a heavy atom, but it turns out EU GD has more unpaired electrons. So with this, we actually you look at the results. So using this the GD as the host materials, we actually can significantly enhance the absorption. This is triplet absorption around the 900 nanometers. So we actually did the ultra fast the measurement. So it turns out this energy transfer between the triplet states and also nanoparticles is on the picosecond time scale. It's a very fast process. So we believe the energy transfer mechanism between the lens organic is the spin exchange process. So one of the you can see once the organic promote this triplet state here, but also it tends to spin flip for the lens ions, spin up to spin down here to achieve this direct triplet generations. So I want to highlight this review article we wrote a few years ago. Here mentioned some of the application here, but I think it's not only for using the lens dopant, rear dopant, not only for up conversion, but also for down conversion. You can actually using this lens dopant strategy. Some of the application shown here. I want to highlight a few emerging applications. I think interesting studies involving this lens ions up conversions. So one challenge for up conversion is I mentioned the efficiency. So how we enhance the efficiency? The quantum conversion efficiency is actually less than 1%. You compare to organic diet, quantum does so almost. So it's very, very low here. But how we can enhance that? So one strategy is using the nano cavity. So here showcase that if you have this sandwich structures, so nanoparticle, these up conversion particles, sandwich between the silver nanocube and the gold film, you actually can achieve significant luminescence enhancement. So I want to show that this is real experiment data. You can see pretty uniform upconversion, more or less. Okay, so we have this single silver nanocube. So this is underneath of the nanoparticle more layer is the gold film. So based on this design, we actually look at the luminescence enhancement. But also, very interesting discovery is we actually can use this sandwich design to control the superluminescent colors. You can see the color actually can tune that. So using this sandwich designs. So this I think this nanocavity design is very, very interesting, not only for upconversion materials, even for quantum dots, perovskite nanocrystals. So we can certainly try other materials. Recently also working with a group from Italy. So my collaborator actually is purely theoretical researcher. So he has a capability to actually fabricate the matter surface matter materials. So one of the challenges, as I mentioned, is the luminescence conversion efficiency, the emission intensity. So this study actually showed very interesting discoveries. If you're using this upconversion material embedded into dielectrical materials, the matter surface, not only we can achieve significant upconversion enhancement, but also we can control the emission direction. So you can see here, this is showing either this direction or we can change another direction at the edge of this matter surface. So that's the most important discovery, actually using we will call this BIC structure, bound in the continuous state. So for the BIC structure is not very hard research areas, but no one actually has reported this edge enhance the direction control. So we've shown that using a combination of the matter surface, dielectric materials, and upconversion materials, we actually can achieve very, very interesting optical phenomena. So I mentioned all this is pretty fundamental energy transfer studies, but I want to highlight several capabilities for biological studies. So all the previous examples more static, means the materials, the nanoparticles is fixed, is not movable. So I want to here show the dynamic, the upconversion in dynamic process, for example, trackings, for example, Brownian motion measurements. So for upconversion particles, so very similar to quantum dots or gold nanoparticles, you can actually use in standard the surface coating. You can coat the particles with a DNA, protein, peptide, small molecules. You can disperse into aqueous solution, biological buffers. You can perform various imaging applications, single particle trackings, photodynamic therapy, so on and so forth. So one idea we thought about is imaging model proteins. Model proteins actually is a very interesting, it's unlike, so model protein in neurons, so they're actually quite interesting because to visualize is actually very challenging. Most of the researchers do the cryo-TM, freeze the sample, and then look at the model proteins. But how to visualize this model protein is performing the particle transfer. So they typically believe the particle is a food. So they take the particle to the somal areas. So there's two types of model protein. One is dynein, the other is kinesin. So the kinesin is removing the particle, the waste, out of the somal body. So to visualize the process is actually already reported using organic dyes, using quantum dots as labels. But those, as I mentioned earlier, so the photostability become an issue. So for tracking, so it means that you're tracking over long distance under excitation. It means that the materials of photostability is a key. So organic dyes is not very stable. The quantum dots, you have a blinking problems. And then we sort of, why not using the upconversion nanoparticles? First, we are shown uptaken into the neurons. Then the model protein perhaps can recognize the particles. So we cannot visualize the dynein, but at least if the dynein pick up the particles from the luminescence, we can actually track the dynein movement. So that's the idea, is model protein tracking. So I want to show one example. So to perform this, we actually use a microfluidic chamber. It's very important. So then we actually can track the model protein using the luminescent labels. So you can see, let me switch on this. So you can see this particle movement. Some of the particles, you can see maybe it's a few particle aggregates, some of the maybe a single particles, but they move almost in the same directions. It goes to thermal areas. So that's actually showing case you can actually really visualize the model protein using this upconversion imaging. So the setup is very simple. So the good thing is, and also it's actually a large area. It's very simple. You can perform this. So this is not, movement is not due to the Brownian motion. So we're already double confirmed that. And also, this is showing the capability. Let me see. So you can see, we actually can see the two traffic, the particle movement to the terminal area and the particle movement to the somer. So you can see that some of the particle stay here, then wait another particle, then so this is double traffic. You can use in this. This demonstration is we want to understand some of the cancer drugs. If the cancer drug have a side effect to the brain, it turns out, so we actually, it doesn't have a direct evidence, but using this kind of study, we can somehow to predict certain chemical drugs. Let me come back here. So here you can look at the particle movement. It's very steady. So that means we believe there's no side effect because the neurons function nicely. So it actually is reflected by the movement of the particles. So in our control, so we have to control the stem cell from the diseased patients. So the particle movement, the pace is very different. It's sometimes very fast, sometimes very slow. So based on this, the imaging results, we can actually perform, for example, data analysis using machine learning to understand if the drug has a side effect to the brain or not. So as I mentioned here, why we're using this upconversion particle, because the tracking, we can have a more data set using the luminescent these upconversion particles. If you're using quantum dots has a blinking. So in between here is once it blinks, we don't know where the particle location, so we can only draw a straight line here. So it means some of the information is missing here. Okay, so this is another application, as I mentioned. We actually can using this the particle luminescence to understand some of the basic questions. For example, the TRPA1 ion channel. So it's very interesting fundamental questions. David Julius and Arden, so they win the Nobel Prize a few years ago. So it's actually studied these ion channels. So one key debate is if the ion channel opens, the sodium can pass through the ion channel. So the debate is if the water molecules will follow the sodium ions passing through the ion channel or not. So this is a very strict questions, but there's actually very much debated because some people believe that once the sodium ions opens, passes through that, water certainly you will go through that. But actually remember the cell membrane also have the water channels. So water actually can go through water channels. So we don't have the direct evidence. So can we design some experiments using these photon upconversion particles? So we actually designed this quite interesting phenomenon you can see here. So in different solvents, so if you disperse the particle in aqueous solvents, you can see the luminescent quenching. Most of luminescence is quenched by water molecules. But if you switch it to D2O, you can see the strong luminescence comes back. So this is based on this luminescent turn on using two different solvents. So we actually achieve this. We can simultaneously recording the channel current and also water signals. We can do this at the single ion channel, single molecule level. It took us a few years, to using this setup, using the micropipette to connect a single ion channel. So then we place the particles within this micropipette. So if the ion opens, so you can see the D12 is actually the solvent in the micropipette. So the water, once it opens, so the water comes out, then the luminescence is quenched. So we can also do the opposite design here. So you can see either the D12 can go inside, the particle actually is within the cell bodies. So we perform the both experiment. Actually, we confirm. So using this setup, so the conclusion is the water molecules, let me come back here, indeed it can go through the ion channel. So we use a luminescent turn-on approach, on and off approach, to confirm that. But of course, this is just for this specific ion channels. So certainly we want to extend this study to other ion channels. And also this TRPV1 ion channel is pretty big. They actually allow the big molecules, micromolecules passing through that. But the water is no direct evidence. Like I said, the water can go through the water channel. Water goes through the ion channel, so you need direct evidence. So we believe we have a direct evidence using the luminescence approach. So I want to spend a few minutes to highlight this optical nonlinearity from the upconversion materials. So that's the number 3, most unique optical properties of lens-and dope materials. Optical nonlinearity, I think most of you already are familiar with two-photon absorption. Two-photon absorption compared to two-photon excitation compared to one-photon excitation. So why we need two-photon excitation? Because we actually can reduce excitation volume to one particular. We can highlight the excitation to a single spot. So the benefit of the two-photon excitation or multi-photon excitations, once we have the small excitation volume, we actually can perform, for example, ultra-fine 3D printing. So unlike it's one photon excitation, so you don't have this focus excitation volumes. You cannot achieve this ultra-fine 3D printing. So I want to highlight this two researchers. Blombergen, obviously he's the father of nonlinear optics, and also Maria Mayer. So she probably unknown to many of you, but she is a very important figure in her PhD study actually is on the two-photon absorption. And her Nobel Prize is actually, she won the Nobel Prize for the discovery of the model for atomic nucleus. I mention her because not only her academic achievement, but also I want to highlight in the early days, she actually spent almost 20, 30 years as a researcher without getting paid. Yeah, so because in older days, even in the States, so the women cannot work. Yeah, so she actually worked at a number of universities, Columbia University, I think also University of California, for 20 years without getting paid. So can you imagine that? So she strongly supports female researchers. And also she's the second Nobel Prize, the female researcher win the Nobel Prize. So photon avalanche is quite interesting. Why I mention this? Because you look at the nonlinearity of this two-photon excitation, then you extend it to 3-photon, 4-photon. You can play with the optical setup. You can achieve nonlinearity index, maybe around 4. That's already ultimate is the limit. So using this lens-and dope particles was reported. You can actually achieve very high nonlinearity, so around 40, 50. So using this, we call this by taking advantage of cross relaxations. So you have to build up high population at this intermediate stage. So one photon excitation, and then go up, once go down here. So you induce this photon avalanche. So eventually one photon excited, and then become two photon immediate, and then 2 to 4, 4 to 8. So you you're going back and forth. Eventually, this is a high, large, more like a long-wave chain reaction. So you can really significantly amplify the population of the excited states at these intermediate energy levels. So the research field has become very hot. Okay, why is that? Because if you can achieve, you can high nonlinearity. You can see the slope here, very sharp. So it means you excite these materials, okay? Initially, if a low excitation intensity, the materials are no luminescence. You increase a little bit, then suddenly the luminescence goes up significantly, just like you're shooting here, luminescence intensity. Then if you further increase intensity, then it goes down. So this is very interesting. Look at the slope here. So one photon here, two photon, you actually increase. So this is the excitation intensity. So the consequence, if you can reach very high nonlinearity, you can actually using this to develop, for example, pressure sensors, force sensors, because the luminescence is very sensitive to the excitation intensity. So I want to give you some examples. This is our recent work. So we realized that if you using interested in using certain type of lens-side hosted materials, we can change the sublattice, the crystal lattice. Then introduce we can modify the crystal field. Once we modify the crystal field, we can significantly enhance optical nonlinearity. So this you can see this is simulation shows the population of the different energy states. You can see clearly see this intermediate energy levels. Significant increase. This is a very sharp increase. So this is a significant optical nonlinearity. Let me show some imaging capabilities. So with this high optical nonlinearity, so we actually can achieve super resolution imaging. So you can see we tested different particle structures. The key materials, elements, LU, used as a host matrix. So we can achieve optical nonlinearity over 156. So this we even achieved more than 500. So that's very, very sharp. Just more like we actually can build up optical switch. So you can see this is quite interesting. With this kind of high optical nonlinearity, we actually can resolve two particles in a very close distance. So conventional optical microscopy cannot resolve these two particles because of the diffraction limit. But using this, we can clearly resolve the particle distance. You can see we actually increase 653 kilowatt. So further decrease, then further decrease. So once we reach the threshold, 603, then become very, very clear. So this is quite interesting. We actually can use in these designs to look at the particles. You compare the SEM image, you can see the particles are 172 nanometers, but our resolution here, this is very, very interesting. Even 91 nanometers. This is beyond the physical size. So this is very unique because in most cases, the optical imaging, you measure the size typically is larger than the real physical size. So this phenomenon is all because we have high optical nonlinearity. Yeah. So I want to show in the optical setup is actually very straightforward. It's nothing fancy. You're using several optical components. You can achieve that. Most important is you have a synchronize the measurement, the power density, and also emission intensities using this spectrophotometer here. So this, I just want to quickly show you. Let me see. Okay, so here I speed up this the measurement. You can see we keep increase the intensity, excitation intensity. Then suddenly, once we reach the threshold, let me show you again here. around, so we measure the 800 nanometer emission. So we keep increasing. The materials is a magical materials. You keep increasing the excitation intensity. Then suddenly you see a huge emission output. So you measure the slope. So this is the nonlinear IRT factors is around 500. Okay? So I mentioned all kinds of some of the bio applications. This optical nonlinearities, nanophotonic applications. I think the future challenge in the field, of course, is integration. So I want to maybe highlight some of these applications along these areas. Light sensing, light field imaging. Because typically when we talk about imaging, detection sensing, we talk about wavelengths, color sensing, intensity sensing. But light has many, many factors, right? That has, for example, polarizations, phase, orbital momentums, so angle detections. So how you combine all these parameters? So how to, for example, develop this on-chip spectrometer is actually the most challenge. So first of all, I want to maybe highlight this light field. Light field imaging is angle detection. So one is very challenging because the conventional light field imaging, the angle detection is using micro lens. So you have to use a lot of micro lens arrays to detect the light angles, incident angles. So we sort of, why not using this color conversion? So you can see many of my research is talking about color conversion. So we're actually using this perovskite nanocrystals, excellent optical absorbers. So we designed, it looks very complicated, but actually the design is used to convert the light angle into colors. Then using the CCD camera, so we can resolve the color index. So that's the idea, is how we can convert the angle of the incident light into colors. So using different, you can see the different orientation of this, we call this azimuth detectors. So based on, so if you have a 2 or 3, if you have just one detectors, so we actually can only detect the light from coming from a certain plane. But if you have a 2 or 3, then we can really determine the color, the light coming from a particular point in the 3D space. I just want to show you, this is real experiment data. So if you tune the color, the angle, tune the angle of incident light, you can see some of the pixel. It actually changes the colors. So using this design, so this design is more like LiDAR, imaging sensor, so we can detect the 3D different depths of the distance of the object. I want to show you this is real imaging. So what we're seeing here is the Singapore most famous Milan statue. So we see the 3D, this color imaging shows the different color showing the different depths of the object seen from the camera here. So we actually can also resolve very fine structures. So this is using the pineapple. You can see the surface morphology here. So this is important, I think, is for imaging. We talk about now the challenge is the resolution. So if you can further improve resolution, I think it could become a very useful tool, competing the confocal imaging. So confocal imaging, you have the X, Y, and then do the Z scan. So you have to play with that. But this one is, once we have this, the setup, we can just take a snapshot. All the 3D information is already there. Of course, the challenge is if you want to using this for biological imaging, you talk about 100 nanometers. The imaging pixel is challenging here. So what are we seeing here? This is actually, I think, the one pixel is, I think, around the 20 or 50 micrometers. So I think the challenge, of course, lies the future research in 3D printing. So this actually, we're actually using the 3D printing technology to print this. Another application of this is actually face imaging. So we actually can use these materials to resolve the wavefront of X-rays. So X-rays are invisible to naked eyes. How the X-ray propagation over long distance. So that's a challenge, it's invisible. So we actually can using this strategy, convert the X-rays into colors. Then we can, this shows intensity mapping here. This shows the face imaging. So from the face imaging, we can look much more the subtle details of the object. So this I think this is another very important applications. Wavefront mapping of visible light. Here's a visible light. And also X-rays using this angle to color conversions. Okay, so I think the future, of course, is integration, as I mentioned. So the smartphone spectroscopy, I think that's the future challenge. Everyone has a smartphone, but we can only take a picture. We can see the colors. But other dimension of the light information is not there. So if we can take this smartphone, not only take a picture, but if we can analyze the chemical composition, spectral information in the picture, that'd be very, very convenient, very useful. You can walk into a grocery store, you pick out some apple, but if you are not sure the apple contains chemicals or contaminated with some chemicals, you can actually using smartphone spectroscopy. Take a picture, you can analyze it. So this become I think the future technology. Of course, there's a many research group is working on in these areas, develop this. So we actually developed this proof of concept, the space method, using we call it stochastic photoluminescence and compressed sensing. It's a combined compressed sensing, then this random photoluminescence. So here, for example, we actually encoded 4 different pictures. You can think about the 4 different wavelengths using 4 different color emitters. So you can see that this is real imaging, the random color displacement. So using this random color displacement combined with compressed sensing, we can decode it 4 different wavelengths or 4 different imaging. But of course, the challenge is the materials conversion and also no crosstalk between this wavelength conversion. So of course, they still have a lot of challenge in the fabrication, in the design here. I just want to highlight this the recent works from other groups, even from the Mojibal Wendy. So he's a Nobel Prize winner. So he actually showcased using the colloidal quantum dots, he actually can develop this spectrometer. Of course, this one you can see actually using each pixel contains different color emitting quantum dots. So the complexity you can see, and also the pixel resolution is still challenging to make larger areas. And also this one does not give you the imaging, only give you the spectral information. Here, this is another work, I think from Zhejiang University. They show they demonstrate a single nanowatt spectrometer, also using quantum dots. So each segment is made a particular composition, so you give it different colors. So this quantum dots covers, this luminescence covers the entire visible region. But this also cannot perform the imaging. So if you want to combine the imaging, spectral information, I mean, how about polarizations, angular momentum? So all these parameters are within one small spectrometer. So I think that's the ultimate challenge, miniaturization. So I think the computer imaging it comes, perhaps can help with this research field. AI, of course, yeah. So I think I want to end my presentation. So I want to share this photo here. So I want you look at this. Try very hard to look at this spot right here. This is the Earth. So this actually taken from the spacecraft fleeing our solar system. So it was taken almost as many years ago. I don't remember. I think it's 30 years ago, I think. So you can see here, this little spot. So it's not a continent. it's not it's just a pixel of scattered light here. So, every day we expose the light, we see the light, but we know very little about light. I think it's still many information. We try to understand light. So that's, I think, my future goals is to understand how the light interacts with the materials. So Carl Sagan once said, look again at that dot. That's here, that's home, that's us. So I see that let us look again with sharper eyes, humbler hearts, and a deeper resolve. So I want to end my talk right here. Thank you.
Thanks, Professor Liu, for very inspiring talks. I think we have heard a lot of works, not just on fundamental science, on different energy migration, energy transfer, but also real demonstrations of technological applications. So I think we now open to questions. Any questions from maybe students first? Okay, yeah.
So thank you, very exciting talk about the nanocrystal and applications, and of course the very deep mechanism. You have shown that you certainly make contributions in solving real problems in different fields, including the electronics, biological, photonics, and of course the chemistry. Actually, to be honest, I think it's very hard for a young researcher to hold all of these different fields. So maybe you can give us some suggestions how to connect these to the real problems and how to solve these problems. Thank you.
Yeah, it's a good question. Actually, for many researchers, of course, it's a daunting challenge because if you want to you have some ideas. These ideas requires a special skill or expertise beyond your research. And that certainly, I think, the mindset is very important. So you need to approach someone who knows the technology, and also someone who is willing to work with you. So that's actually very important. You have to convince, for example, we have some of the work I never mentioned that involves other research, biomedical electronics. So the student, I think, even student or postdoc, so you really have to, go down to another level, convince someone that you can really work with you. So I think it is because every time, I travel a lot of places. Sometimes I run into someone who has expertise in, for example, optogenetics. So I talk to the professor, and professor talk to students. Typically, I assign the students, the student contact. So actually, we published a paper, didn't mention it, published in Science in optogenetics, using upconversion for optogenetic study. So that's actually an interesting story because the whole collaboration is I was invited to give a talk in Tokyo, University of Tokyo. So the student came to the airport to pick me up. And then the student shared his research. His research is very different from this upconversion. But I introduced my research. So we actually talk about this upconversion, for 30 minutes during the, in the subway. So then he became very interested. So after I went back to Singapore, and then he still contacted me. I actually had some discussion. We actually assigned the students and then work with that student. So after he graduated from Tokyo, University of Tokyo, he actually became a postdoc in RICON, the Brain Research Center. So that research is very different from his PhD. His PhD work, he works with AIDA. AIDA, everyone knows that is a supermolecular chemist. So he completely switched the field and then jump into optogenetic field. So he actually worked with my student. So we collaborate and developed this very interesting upconversion, optogenetics demonstration. I think so. That means so. I think my advice, of course, is you really open your horizon. So it's very important. Many occasions is researchers seem to limit it to his or her research field. But I think once you opens your mind, I think perhaps there's many opportunities. Yeah.
So don't just focus on what you are doing. Also, expand your skill set. Any? Okay, I'll check.
Thanks. Enjoyed a lot. Very inspiring talk. So I work on a display actually and use this kind of nanoparticles like in various devices such as color conversion or electroluminescence or different photonic components like that. So very interesting talk. So I am interested in two projects when you talk about this remote control of the frequencies, right?
Yeah.
So you can control all the red, green, and blue using the IR lasers.
Yeah.
So the question is, some of these long wavelength, the long pulse duration that you mentioned, it is somewhere in the range of milliseconds.
Yeah.
Right. So, and some of them are in microseconds.
Right.
So what exactly is the factor that defines this pulse duration? That's question number one. And question number two is how stable they are. Like means if I want to reproduce this color again and again, how reproducible they are.
Yeah. So the first question is, this because the lens dyes typically give a very long luminescent lifetime. Yeah. So I think the control is, so you can see the core shell structures. So this pulse duration control is, so we actually control the energy transfer within different layer of the nanoparticle structures. That means we control, so the energy transfer, for example, can transfer from YB to TM, YB to ER. So we know that ER gives one color, TM gives another color. Okay, so this already, this answers the second question. Very stable. So unlike the quantum dots organic dyes. Quantum dots, of course, are pretty stable. Only problem is maybe the blinking. Organic dyes is quenching, bleach. So the upconversion, this laser dopants. So the luminescence, the wavelength, essentially is fixed. If you change the size, if you add some impurities, but if you see the emission, always that particular wavelength. So either ER emission or TM emission. So the questions, I think the challenge is how you control which ion emit. So because we tune the color. So that's why for laser ions, crosstalk. This crosstalk, we call this as energy transfer between laser ions. If we can control that, for example, using the core shell structures, so we can place the ER in the core, then the TM within another shell, so we can really control. So that's how sometimes we're using CE as energy migrators. So once we can achieve that, so this becomes very easy. Actually, it's tunable and also very stable. You can repeat this many times, no problem. Yeah.
That's very interesting.
Yeah.
Very exciting. So in this scenario, the quantum yield is as you mentioned before, it's still very small. So is there any way to increase it at least to a certain level where you can use it for displays?
Yeah, that's actually I think the holy grail in the field. So I showed you several strategies. For example, nanocavities, BIC structures. So those I think but of course the enhancement, we can see very strong luminosity enhancement, but the quantum yield is still very low. But that's actually, in terms of the brightness is already, so I didn't bring the near laser. So if I bring the samples, I can show you. It's actually strongly visible to the naked eyes. Then I think with AI, machine learning, I think the ultimate challenge is finding the right materials. If you can find the right materials, maybe, just like the perovskite materials, people realize that this is excellent photo absorber.
Right.
Yeah.
Mm-hmm.
Yeah.
All right. Actually, I have many questions, but
okay.
let me give some chance.
Yeah, sure. We can discuss later. Yeah.
it's interesting to show that you can use that to sense the transport of water molecules.
Yeah.
So I'm wondering, like, the effects of the solutions on the optical properties of those nanocrystals. Like, is there any potential that will aggregate and affect the properties?
Yeah, so that's actually a very good question. But the good thing about these lensable particles, even the aggregate, the luminescence is essentially unchanged. So it's like quantum dots or gold nanoparticles. Gold nanoparticles, if you aggregate, you see the surface plasma shift to the red, right? Very sensitive to the particle size. So even the particles are dispersed in aqueous biological buffers. So even you see the particle aggregates, but luminescence is very consistent. You will not see the quantum confinement effect, the particle size effect absorbed from the quantum dots. Yeah, so that's actually the good thing for our research. So it's these particles has minimal influence from the environment. Yeah.
Wow, that's great.
Yeah.
And another question is about the application side, because usually when we use conversion techniques, we use it as a detector because it converts those.
Sure.
It's very expensive to detect the infrared light, but now you can just use visible light. So have you explored those applications?
Yeah, that's because of time limit, I didn't show it. So we actually designed the several strategies to detect even in the middle infrared region. So using up conversion, we actually can detect, we can actually image in middle infrared light. Middle infrared, we're talking about 4 to 10 micrometers. So now we try to extend it to maybe terahertz. Yeah.
Wow, so is there any limit that you can detect? So, or you can just turn it to absorb any wavelengths you like?
Well, it really depends on. So, for example, I didn't mention the X-ray sensing. The particles also, because you look at the luminescence, this lens I show one slide, I show the energy levels. So they have many 3,000 energy levels. So some of the energy levels have very small gap. Some is a relatively bigger gap and large gap. So those are very small gap because that's the orbital splitting here. So that's if you look at the wavelength, the energy gap. So then you compare the wavelengths. Some of the gap is, for example, is you can match the wavelengths of the middle infrared, even terahertz. So of course the thermal stimulation become an issue here. So then you need to perform the experiment at the ultra low temperature. Yeah, but certainly it is possible. We already done some results, experiments in this area. Yeah.
Yeah, I think thank you very much. I think I'm coming here to learn. Actually, it's also learn a lot. So my question is related to the causal structure.
Sure,
when you use different laser, you see the different emission.
Right.
And my question is that have you tried to arrange the different order of this different layers, so that you see what's the mechanism for the energy transfer?
Yes.
So you need to have a mix. I'm not sure. I think just simply it's interesting to know that when you use a different laser, you see the different emission, and then if you advance in them in a different order, what kind of result you have seen?
Yeah. So we play with the causal structures. So different layer, we actually play with the dopant concentration or combination. So it's a lot of work. Actually, we have done all the experiments you mentioned. Yeah. So then we try to solve the mechanism issue here. So
because if you get insight, and then I'm sure will be like what you expect, that some sort of how to tune the core structure in order to achieve what you want.
Sure. Mm-hmm. Yeah, we actually did a lot of simulation. Yeah, the simulations, numerical simulations have become the key part of our research, especially you talk about there's so many energy levels you can perform
because the mechanism of energy transfer.
Yeah, so many energy transfer pathways. So that's why the numerical simulations can really help us to understand the energy transfer between not only between the lensite dopants, also between the ions in different layers. Yeah. So that's actually very systematic studies. Yeah. Yeah.
Okay. Maybe I will also ask a few questions. Take
Sure.
this opportunity. So I have also many questions, but I'll just ask some quick questions. So first question is, why is the water quench the up conversion, but diluted water not?
Yeah, I think it's an interesting phenomenon. And many other groups already observed because the up conversion is actually. So first of all, you need to understand why these lensite doped particles only lensite doped particles give a photon up conversion, not a lensite complex. Okay, if you think a lensite can perform, why not a lensite complex, right? So the key difference is the lensite complex using lensite organic ligands. These organic ligands, have strong vibration, but for lensite inorganic particles, so they have pretty rigid lattice. So this rigid lattice actually protect this excitation energy. So once the absorbed 18 nanometer, you have this excitation to the intermediate states. So that's give you the protection here. So the water molecules, remember most of the up conversion I mentioned energy migration. So migration happens within the particles. So it means if the particles emit light, so not only the core, but actually can transfer to the shell. Mm-hmm. Shell layer also have the emitters. Yeah. If the water contact this emitter, the water has a strong vibration frequency. Yeah, so it quenches luminescence.
I see.
So D2O, no. D2O is very interesting. D2O has a very, very low photon energy.
So all the experiments you have to be done in non-aqueous, or when you do the up conversion, if water is a problem, what's the solvent?
Oh, that's a good question. So typically we actually grow a inert shell. Okay. So if, for example, using sodium yttrium fluoride, so that's already
another shell.
It's like a recipe.
That's a protection.
Yeah, yttrium. Look at yttrium is very, very good in inert shell. Right. You coat it. You can actually stabilize.
So when you talk about energy transfer mechanism, you seems to mention is a fret mechanism. In some cases, I don't recall. But when the energy migrate from lens line to lens line, is it a fret mechanism or is it dexter mechanism? So that's question one. Question two is when the energy transfer between lens lines and say tyrosine, is it also a dexter mechanism or what's the mechanism?
Yeah, so it really depends on the distance. The fret force. Resonant energy transfer, so it's a long distance. Dexter is a short distance. Right. I think it combines, the energy transfers has both. So it depends on the distance. It's a combination of both. Okay. But energy migration is a special case, energy transfer. So this is unlike a FRED. So FRED they typically don't accept it. Right. So this energy migration, like I said, is more like you have this. The GD ions within the lattice, so they are very close distance. It's more like a resonant energy transfer through the GD ions. So the GD ions, the electron energy level is about 4 electron volts. Right. So they are pretty stable. Even water will not quench that. So through the GD, I think it's elastic energy transfer. Yeah. So one GD transfer to another GD.
More like a resonant.
Yeah, resonant. Then go through the lattice. Of course, it's a random process. It can go through here, there, then come back. Yeah. Or this then go different directions. Yeah. So we have to, once again, do the simulation. Yeah.
But I think that the energy transfer between lens line and the organic molecule, from your mechanism, looks like you have a spin exchange. So that's more like a dexter mechanism.
If on the particle surface, if you have nanoparticle donor, the organic dye on the surface,
they have to become very close in order to do that.
But in most cases, we think once the energy transfer to the particle surface, then to the organic dye, we think it's a FRED.
You think it's a FRED?
Yeah. Then it's actually a FRED. If it is very close distance. Yeah.
Okay. I think.
Yeah.
I'll read more.
Question.
Okay. Yeah.
So, sorry. I was reading through your essays again, and then I realized that the matter nanocrystal allows for localized reference plasma resonance, and it can increases the upconversion critically by a lot. But then I realized that you do it source state, you sandwich it, and you said it before because in solution form you can't really see that.
Right.
Is it because of mobilization, or is it because of concentration ratios?
You mean the sandwich structure within the solution?
Yeah. So what if I increase by the metal nanocrystal by a lot? Then does it work, or?
Yeah, actually, interesting suggestion. We have never done that, but I think perhaps in most cases the nano cavity is demonstration is in the solid state. Yeah, maybe I think a solution, just I mentioned, is of interference.
Okay.
So maybe quenching the luminescence.
Okay, that makes sense.
Because any solvent is a strong absorber energy. Yeah. So perhaps So that's why. So we always do this in the solid state.
Okay. Thank you.
Any more questions? Otherwise, we'll conclude. Let's thank Professor Liu again for a very inspiring talk.
Thank you. Yeah.
Thank you very much.
Thank you.